afternoon. My name is Shakoya Mack, and today I will be presenting on the role of ADRM1 in breast cancer. Why does this matter? This matters because breast cancer is a leading cause of cancer related deaths worldwide, especially in female patients. Because, yes, males may also get a mutation in the breast tissue which causes breast cancer. Triple negative breast cancer, which is termed as TNBC, is highly aggressive. And it's highly aggressive because it lacks the necessary receptors, hormone receptors, and proteins that are normally required for treatments and that make treatments options more available. So, those receptors are the estrogen receptor, the progesterone receptor, and the HER2 protein. And when you lack those three things, that makes your treatment plan harder because of there's a lack of availability, there's a lack of targets. Okay, when I say that it has a higher recurrence rate, I mean that in cancers overall, we have what you call a 5 year survival rate. And with this 5 year survival rate, after you've been treated, um, normally they give you about 5 years to see if you will get the cancer again or develop some type of new cancer, or will the cancer come back and metastasize. So a higher recurrence is in TNBC. Within 3 to 5 years, that cancer has progressed severely to stage 3 or stage 4 after you've thought that you've been cured or treated. And a poor survival rate. So what I mean by it has a poor survival rate is because this is the most aggressive form of breast cancer, the likelihood that a patient will survive this is less likely, especially um, when it comes to higher ADRM1 levels. African American women face disproportionately higher mortality rates. Black women receive a breast cancer diagnosis around 40% more than their white American counterparts. What contributes? TNBC shows EMT and basal like features driving invasion and aggressiveness. So, what I'm saying here is the epithelial mesenchymal transition is basically when you go from a healthy, normal cell, and this cell is mutated or defected, and it grows into like this snake-like snake-like features. So basically, what I'm saying is the cell membrane and the conformation of the cell changes. So instead of it being localized and anchored when it's in this snake-like form, or like it's poorly differentiated, then that's how it's able to get into the bloodstream and travel to different parts of the body. And in triple negative breast cancer, particularly, it um, likes to go to the brain and the lungs, opposed to the bones in other breast cancers. BRCA mutation, BRCA stands for breast cancer gene, yeah, breast cancer gene. And there are two types, BRCA1 and BRCA2. And normally, its normal function is to help like generate healthy tissues, healthy breast tissue, and BRCA1 also plays a role in cell division. So typically, it acts as a tumor suppressor gene or it acts as a tumor suppressor. But when the BRCA gene is mutated and it has a high mutation burden, that's what causes the genomic instability. And when you have this mutation, it is also like family linked. So if you have somebody that's in your family that had a BRCA mutation, it makes it more likely that you or someone in your family would get this mutation. And ADRM1 levels are also associated with the BRCA mutations. Tumors show immune infiltration but evade the immune system. So here we talk about the immune cells, specifically T cells and PDL1, which stand for Programmed Death Ligand 1. And basically, what PDL1 is it is on the cell surface, and what it does is prevents T cells from attacking healthy cells. But again, TNBC hijacks this mechanism, and it uses the mechanism against the cell. So instead of it attacking the breast cancer cells, the breast cancer has turned this mechanism off, essentially. So, or underregulated. So tumor suppressor genes like P53 or the T cells, they can't even help out your immune system because they're essentially being tricked to thinking that you don't need it. Um, Triple negative breast cancer relies on glycolysis and stress pathways, and because it uses glycolysis and the stress is what upregulates the ubiquitin proteasome system, and that's why it says increased UPS dependence. Here is a overview of just me showing that your white counterparts, yes, they have a higher incidence. So we'll look at the female side first. If you look at the solid red line where it says new cases, this stands for our incidence. And under the white, you see we have 423.1. And then if we look at the Black American, it's 397.8. So again, that's emphasizing that the incidence is lower in blacks. But if you look at the dashed lines, the death rates or the mortality rate, you see that the Black American is leading in the death rates opposed to the white Americans. And you will see that again if you look on the male side, which is in blue. Factors that contribute to high mortality rate of African American women: a family history, which again we spoke about the BRCA gene or the breast cancer gene, delayed follow-up appointments, lack of medical facilities and trained health professionals, and lack of clinical trials for non-whites. Breast cancer subtypes and triple negative breast cancer differences. So, breast cancer is not one single disease. Each subtype of breast cancer comes with its own characteristics. So, luminal A and luminal B, they are hormone positive for the most part. And what does that mean? When you have hormones on your breast cancer cells, or the receptors are present, that allows you more treatment options. So luminal A breast cancer is your best chance at surviving breast cancer, and especially past 5 years. Even if you make it to stages 3 and 4, you're still better off opposed to somebody that has triple negative breast cancer stage 1. That's crazy to think about. Luminal B, it's a little more aggressive than luminal A, but you still have a high percentage. So like around 40% of people get luminal A, around 20% luminal B. Then you have your HER2 protein, which stands for human epithelial receptor 2, and it has its own set of therapies. Triple negative breast cancer. We've stated before or earlier in the presentation that it lacks the necessary receptors, which are the estrogen, progesterone, and HER2. And it has high mutation rates and greater adaptability. And it is more likely to metastasize to the brain and the lungs rather than. Again, this is just another overview showing you that luminal A has the best prognosis, while triple negative breast cancer has the worst prognosis. Um, Ki67 is just a biological marker depicting the rate at which cancer cells are dividing. And normal like would be in between luminal A and luminal B as far as severity goes or survival. The big idea. The big ideal here is that cancer cells or cancer, especially in TNBC, relies on a ubiquitin proteasome system, which plays a role in protein balance and protein homeostasis. ADRM1, which is a ubiquitin receptor within the proteasome, represents a key regulatory point that may serve as a potential therapeutic target. And when we think about this slide, again, we're thinking about what is the cancer relying on to survive? And the answer to that is relying on the ubiquitin proteasome system to help it survive. And so one mechanism that it uses is generally like, okay, so the ubiquitin proteasome system again is for protein degradation, and it uses the ubiquitin tag. Okay, so normally, oh, let's talk about in cancer first. So what cancer does is it upregulates the proteasome, causing it to work more. So when the proteasome is working more, it is essentially clearing out all of the like the tumor suppressor cells or just your more healthy cells. And if it's clearing out all of the cells that keep cancer in check and it's sending them to be degraded, or it's tricking the system to degrade these type of cells, that means that the cancer cells are able to proliferate more. Okay, and so that's how it uses this ubiquitin proteasome to its advantage. Metabolic stress and obesity. So basically, excessive nutrients increase metabolic demand. Okay, and this leads to oxidative stress and inflammation. Right, so with this higher metabolic demand, it increases your levels of oxidative stress, or it increases your levels of reactive oxygen species. And basically, these are just harmful molecules, or what do you call it, like free radicals that are floating around and they damage the cell. So they damage the integrity of the cell. You can call it like a biological rusting of the cell, if you will. So yes, the more you eat, the more you could be feeding your cancer, and the more like the body has to work. So all of that plays a portion in it. So here, under the ubiquitin proteasome system, we can talk about the pathway that it takes. So we have three key enzymes within the ubiquitin proteasome system. That is E1 activating enzyme, and this enzyme uses ATP to help it activate, and it binds to a ubiquitin and the substrate. Next, we have our E2, which is our carrier, which binds to E3, forming a complex, ubiquitin complex, ligase complex. Okay, so what this complex does is it actually recognizes the proteins that are tagged with ubiquitin. So again, I told you it was a regulated system, right? So with it being regulated, it's not just going to attach itself to any cell that it sees available. No, it's only going to attach itself to the proteins, or to the proteins that are tagged with polyubiquitinated chains, because the more chains that you have, the more affinity that it has to the proteasome to be degraded. The less amount of ubiquitin on that substrate, it has the potential to kind of fall off, or maybe not fall off. There are other enzymes and mechanisms that work within this, but to sum it up, if it has less ubiquitin, less ubiquitin molecules, then it may not get degraded. It may not make it to the proteasome. In here, I'm just showing you a picture with the enzymes, and just showing you how with the protein substrate and how the ubiquitin tags it, or attaches itself to it, and how it goes through the proteasome, and how it reuses peptides. Ubiquitin proteasome dependence and role of ADRM1 in triple negative breast cancer. Cancer cells produce large amounts of misfolded proteins due to rapid growth. And that's just simply what it is. Other things that can cause proteins, again, just the overproliferation of proteins being made and replicated is one way that these proteins can misfold. Um, Within the transcription, if anything went wrong, or if your DNA somehow got damaged, or like high heat levels, those are things that contribute to the proteins misfolding. And under normal conditions, a cell would die. But that's part of the adaptability when you talk about triple negative breast cancer, because it still thrives. And furthermore, triple negative breast cancer prefers to, again, to use the glycolysis pathway, because the energy is quicker. Okay. This stress makes TNBC cells highly dependent on protein quality control system, like the ubiquitin proteasome system. Um, ADRM1, which sometimes is known as RPN13, and RPN13 stands for regulatory principal non-ATPase 13. And so basically, it's just describing that in that portion of the proteasome, because it sits in the proteasome, that portion of the proteasome does not use ATP to operate. And again, it is a ubiquitin receptor, and it also acts as a docking station for the ADRM1. Okay, this is a picture of the proteasome. The whole thing is called the 26S proteasome, but the 19S is the regulatory particle, or what you would call the lead, and through the 19S regulatory particle, or RP, is where the polyubiquitinated chains and substrates will come in, and again, they're going to be misfolded. So at this lead, before they come in, they are straightened out, to sort of say. And then the 20 is proteasome, or your proteolytic site, is where all the proteins are being broken down at. And at the bottom, or the base, the 19 is regulatory principle. It's where the peptides will come out at. ADRM1 in metastasis and therapeutic potential. Again, ADRM1 plays or does more than regulate proteins. It also has roles in cell adhesion, migration, and invasion. Okay, altered ADRM1 expression can increase tumor aggressiveness, and interaction with the tumor microenvironment. And targeting ADRM1 may help disrupt both protein regulation and cancer spread. So we are truly looking at ADRM1 and seeing how can we use this protein to disrupt these cancer cells, and within the proteasome, without disrupting the whole proteasome. Because within the proteasome, yeah, there are cancer cells that we want to clear out, but again, there are normal cells, that need to be processed as well. So that's why we are targeting ADRM1 and not the whole proteasome. And combining ADRM1 inhibitors with anti PDL1 immunotherapy may improve um, its treatment response. And proteasome inhibition can increase antigen presentation, again, helping the therapy recognize the cancer cells, and that way is able to target only the cancer cells. Experimental approach. Test ADR1 inhibitors in breast cancer cell lines to assess the effects of viability and protein stress. Um, and a few ways that we can do this through PCR, we can do this through ELISA, we can do this through Western blotting. Use of in vivo models such as mice models. They will help us be able to measure the size of the tumors and see if the treatments are working, and to just see all of the physiological things and even the physical manifestations of triple negative breast cancer, and using ADRN1 inhibitors, or proteasome inhibitors targeting ADRN1. The impact. This is just re-emphasized, kind of like an overview. Targeting ADRM1 disrupt cancer cell survival, being protein regulation. Okay. Triple negative breast cancer. Although therapies have been improved in general for breast cancer, there's still a lot of work to be done with triple negative breast cancer. Um, this study incorporates metabolic stress and molecular disparities for personalized treatments. And we hope to improve the outcomes of high risk populations such as the. In conclusion, targeting ubiquitin receptor ADRM1 will disrupt cancer survival, and this will offer personalized strategies for this type of aggressive cancer, and it should close in the gap of the mortality rate in high risk patients.